It's a great pleasure to be able to introduce our first speaker of the afternoon session, Ryan Kelly. Ryan's been a longtime leader and innovator in the field of single cell proteomics. He's developed a number of different innovative sample preparation methods, analytical approaches, and he's used those tools to come up with really great data sets that have really moved our understanding of single cell biology forward. I was particularly excited by a paper he published last year, taking individual motor neurons from patients afflicted with ALS and looking at the proteomes of neurons that have pathology and do not. The sort of data set that was you could really only dream about a few years ago. So really incredible work. And I think in the context of single cell proteomics more generally, one of the things that's been very exciting in the field is to see gains in throughput. And in that regard, Ryan's title, which you can see here, Single Cells in 5 Minutes or Less, I think is a really exciting title, and I'm sure it'll be a great talk. Please welcome Ryan.
All right. Thank you very much. All right. Well, I'm really appreciate you acknowledging that we did some biology because there won't be any of that in this talk. So I loaded my slides onto a USB drive to load onto this computer. And let's see. I don't want to. Okay. Okay. And I found a PowerPoint slide with this in it, and it's from August of 2016. So it's like an old USB drive that I just pulled out of the pile, and all the files in there were from 2016. And I saw this, and I was like, oh yeah, 125 cells, and we saw a chromatogram. I have no idea what we identified, but we were clearly excited enough to put this screenshot into a slide in August 2016. Okay, so I am involved with this company, Micromics Technologies, and I just have to show this slide to show that, yes, we make these really great columns and emitters and heaters and things like that. And we're working on an LC system that should be very, very flexible. Okay, so as an outline, I said no biology in this talk. Pretty much, I think I'll stick to that. Okay, so simple sample prep for label-free single-cell proteomics. We are very interested in at least TMT based single cell proteomics, and we are so bad at it that I think we need to borrow Andrew for a weekend or something and get us up to speed. So everything we'll be talking about here is label free. Then we'll have talk about our multi-column nano LC system that gets us to a throughput of 288 samples per day, or one analysis every 5 minutes. We'll look into some trap columns versus direct loading or trap columns, see how much we're losing there on the trap columns, because that's been a concern of ours. The impact of post-column dead volume on single-cell proteomics. I'm really stepping in it here, doing some comparison between the timsTOF Ultra and the Orbitrap Astral. And then we'll look at going to higher throughput, like 1,000 samples per day, label-free, potentially. And then how we're working to automate the data analysis as well. Okay, so getting started, sample prep, and this is nothing really new here, but just kind of a reminder that as we started with the nanopots approach, it was a lot of steps. Adding a reagent, incubating, putting it back into the system, adding the next reagent over and over and over again, like maybe 5 steps. And so this is a manual process. It involved a homemade robot, homemade slides. And so things have gotten a lot easier for sample prep, at least in the label-free space. We've kind of collapsed all of these steps down to ditch reduction, ditch alkylation, and since we're using this rapid trypsin from Promega that's stable at 70 C, that's also a good temperature for lysing cells and things like that. So we can essentially, one pot, one step, 1 hour, same proteome coverage, and we're doing this in the sometimes in the TCAN Uno or in the Cellin-1 system, we can dispense, but we've kind of favored well plates rather than our old custom microfabricated slides, because the bottom of these well plates, the surface contact is similarly small and it works just fine. So we really don't see any compromise in doing this over a nano well. And with the exception that, at least for spatial proteomics with our laser microdissection setup, that is hard to shoot all the way. The collector is upside down and it's hard to shoot all the way to the bottom of that. So for that, we still use nanowell chips. All right, so yeah, that's basically get a cell in, dispense a reagent, incubate, do a low flow separation, and a high resolution mass spec analysis. This I've also been talking about for a little while. So it's our 2-column high throughput nano LC platform. And it uses a storage loop concept similar to what EvoSep uses, but there are 2 LC columns. So you can see if we fill a storage loop with sample plus its gradient, we can just use an isocratic pump to push that sample through the column, while at the same time we're regenerating the other column. And at the same time, we can be pushing from a binary pump through the trap column to load the next sample and gradient into the storage loop. And while that's happening, we can be queuing up the third sample and getting that ready to run. So besides 2 columns, we've got kind of 3 samples moving the system at any given time. And so Xiaofeng started kind of the original geometry of this, and Chow more recently has reduced this down to a 5-minute analysis. And you can see this is running actually on a LTQ, not even an LTQ Orbitrap, just an LTQ, because why would you want a good instrument collecting boring data for a week, right? So, so what we have is we're just looking, here's 1, 2, 3, 4, 5 peptides eluting from the system over the course of a week, and green is eluting from one column and blues from the other column. And you can see they don't perfectly line up, but it's kind of easy to kind of align those after the fact. And we've run here 2,000 analyses with the same setup, and we've gone to about 6,500 working on the Astral with the 2-column setup without changing the LC columns. So, if you're going fast, you need to be concerned about if you're having a lot of sample carryover from one analysis to the next. But since we can, with this setup, we can sort of selectively elute the good stuff here and then direct the rest to waste. So the super hydrophobic stuff doesn't ever see the LC column. And so you can see we can elute in from column 2 here, and then from column 1, that didn't have any sample on it before anyway. So that is clean, but then going back to column 2, it's also clean. There's just nothing eluting there. So that's working well. Working with this system, Lavender Lynn, who's one of 3 women in my group that had babies in the last month, which is great. Here's 2 of the new lab members here. So she has kind of done a lot of the data analysis with the single cells that were run at this 288 samples per day. And you can see if we sample just Pierce Gila Digest wet from a well, we get about 4,300 proteins detected. And then if we allow that same sample to dry and then we reconstitute it and analyze it that way, and I'm not sure, maybe we didn't have DDM in the system for this, but we took a big hit, and that's kind of how we also analyze our single cells. But still, about 3,000 proteins per cell every 5 minutes is pretty good. These are looking at CVs of technical replicates here and biological actual single cells here. So it's good that single cells aren't all the same, or there would be no point of single cell proteomics, right? And this is very unexciting, but I guess we actually had a nice biological application in there, and something went wrong. So it's like the treatment didn't happen or something. So we're like, they don't look different. So something we have to work on. So if you want to test your quantitative performance, often in the proteomics field, we make these mixtures of human, yeast, and E. coli samples, and then we play with the ratio of yeast and E. coli relative to human. And so again, this is in that 5-minute analysis. The human, the yeast, and the E. coli together make about 385 picograms total protein, 5,900 proteins per sample. And this is just, what part is the human, the yeast in their different ratios, and the E. coli in their different ratios. And the observed to expected ratios look pretty fine here with that. Okay, so moving on to trapping columns, how much sample are we losing? So you can do direct loading onto an LC system using a 6-port valve, but the sample loading flow rate is restricted by the back pressure of this column. So one trick that like the Vanquish Neo does is just go to really high pressure for that. But I'm like, if we have a lot of pressure accessible to us, I want to use that for the actual separation too. Or you can work with a trap column. And our 2-column system's pretty dependent on trapping, where you have essentially a short LC column here that has a much lower back pressure, so you can do faster loading. And it does keep some of the nastier stuff away from your analytical column, which is nice as well. So this is looking with 10 nanograms HeLa, looking at no trap versus a trap column, and we really see quite similar coverage, quite similar hydrophobicities across those. So no deal breaker there. And then we looked at different trap columns themselves, like what do we make the trap out of? These could be 5-micron Dr. Maisch particles, 3.5 micron waters, symmetry particles. And you can see the peak width distribution here is pretty similar. So you have no trap in blue, and that 3.5 micron one is almost identical. And we get just a little bit wider peaks with these other two. And then we can analyze 200 picograms or 10 nanograms in 1 microliter or 10 microliters, and that all works quite well. We were surprised that we could, after prepping, dilute a single cell size sample into 10 microliters and not suffer appreciably from that. So that worked well. And then here, this is just looking at proteome coverage with these different particles and different loading flow rates. 10 microliters per minute, which would allow us to do really fast sample loading. Doesn't seem to hurt anything there. Okay, so next little vignette here is post-column dead volume and its impact on label-free single-cell proteomics. And as Jared from Ion Optics mentioned, they've been screaming about the benefits of an integrated LC column for a long time without like a union and a separate emitter. There are benefits to having a union and a separate emitter, which is when you bump that emitter into something, you just throw the emitter away and you still have a good LC column. So there are reasons to want an integrated column and reasons to need to be especially careful there. So what we've been doing for our 2-column system is essentially in front of the thermo systems is modifying the flex source with this 3D printed adapter that allows us to point 2 columns at the inlet of the mass spectrometer. And then we flip which one is this electrospraying and which one receives the high voltage. And that works well on a thermo system, But on our timsTOF, what do we do for 2-column LC? We can't, right? Because we've got the captive spray source there, and so we haven't had a good way of doing 2 columns into one mass spectrometer. So, okay, so here's a photograph of an integrated emitter. The packing material goes to about here in our case. Then we have a little frit, and then we leave it open to minimize the back pressure there. A column with a separate emitter would be something like this. Here's your column, here's a union, here's the emitter, right? And then for 2 columns with 1 emitter, we thought, well, maybe we could put a 4-port valve after the LC separation. So column 2 would go here and might be directed to waste while column 1 is spraying. And then we would switch the valve. So column 1 effluent's going to waste while column 2 is going to the emitter. So that's what we were interested in doing, especially to get in front of the timsTOF. And so, and by the way, going from an integrated to or a non-integrated emitter to an integrated, up to this point, we had not seen much of a difference. This is with Gary's, where's Gary? There he is. So this is from a 2020 paper where we used a pico clear union and polished the column and polished the emitter. And we didn't see any change in kind of peak widths with or without an integrated emitter or a separate emitter. But these peaks were about 30 seconds wide. So you're not going to notice a big difference. But now as things get faster, the peaks are narrower. And so this is what we're seeing. If we have a separate emitter versus a valve with a 150 micron column operating at 1.2 microliters per minute, we see a little bit of increase in our peak width, and a little bit more when we go to the 100 micron column. And these are all 15 centimeters long, so the linear velocity and kind of the separation efficiency should be the same. We're just adjusting the flow rate by changing the column diameter. And at 75 microns, we tested an integrated emitter, a separate emitter, and a valve after the fact. And you can just see that we get narrower peaks when we use an integrated emitter versus a separate emitter. And about 2 seconds wide with an integrated emitter, maybe 4 seconds, 3 to 4 seconds wide. And so it doesn't seem huge, but this is what in terms of proteome coverage, a couple of features here. So we are going, this is 2 nanogram HeLa digest here, 10 nanogram HeLa digest on the right. And then in blue, this is going from a 5-minute to a 10-minute to a 20-minute gradient with a valve, or in orange with a separate emitter. And you can see it really is fine if you're working at 1.2 microliters per minute with this large column. It's also interesting that you can go from a 5-minute gradient to a 20-minute gradient, and the proteome coverage is very similar, right? But even going down to a 100 micron column operating at 530 nanoliters per minute, if you have a large sample, they're still not that different. But as you go to smaller samples, there's clearly a benefit to not having that extra post-column dead volume imposed by the valve there. Still interesting that the coverage is fairly flat. And then this is with a 75 micron column. Now we're looking at single cell sized samples, 10 cell size samples, and 50 cell size samples. And the smaller your sample, the bigger the benefit of keeping those peaks narrow and tall, right? A skinnier peak is a more intense peak. So we're getting about 4,000 proteins quantified in 5 minutes with this system. And now the last one is going down to 50 micron, and the difference is especially huge from a integrated emitter column to a non-integrated emitter column. And now we're up almost to 5,000 proteins every 5 minutes for that. So, and then you should rightly be concerned, like how much you might be undersampling at some point, right? And that would lead to really large CVs, right? But they're actually not too bad. So this is for the 50 micron column, 200 picograms, 2 nanograms, and 10 nanograms. Even at 5-minute analysis, you know, if you have a larger sample, your CVs are median CVs are less than 10%, and they're in the 13 to 15% range here. So that's for a 5-minute analysis. So it looks like we're able to go pretty fast. Okay, so now let's get political, right? Okay, so so I think there's really good things for the community in general here, and for both Bruker and Thermo here. And the huge caveat is that we just used the same acquisition setting all the way across this, and same acquisition setting all the way across this. I think it was a 150 millisecond ramp time here, and this was a 4 Thompson isolation window on the Astral. But you can see, maybe you can see that the timsTOF has better coverage that we saw under these settings at 10 nanograms than the Astral, and then the situation is reversed at 0.2 nanograms. But not a night and day difference by any stretch here. It's also interesting to see in both of these that, you know, you can go 5 times more sample and your proteome coverage increases by this much, right? And on both instruments. And that's something that, this is a Thermo poster, I think from last year's ASMS, where you can see that, right? So going from 10 nanograms to 20 nanograms, you get like 4 extra proteins identified, right? Again, looking at CVs for both instruments, they're great all the way across from 200 picograms to 10 nanograms. And it's interesting that going longer in our hands for both of these systems just does not provide tremendous gains in what we're able to quantify. In fact, with the timsTOF Ultra, we're maxing out at 10 minutes, and that would be 144 samples per day. And it's essentially flat up here for the Astral as well. One area that we're seeing is kind of the linearity as we go from 0.2 nanograms up to 10 nanograms. Our signal on the Astral goes up like exactly 50 times, and it's just super linear as you change the sample input. It's a little bit less so. This is still pretty good, 0.95 here for the timsTOF. But if you drop that 10 nanogram and just look from 200 picograms, 500 picograms, 2 nanograms, the linearity is improved on the timsTOF, and that is indicative of some saturation that's happening. I'll show you that. Okay, so this is just CVs. Again, these are pretty good CVs, even at 5 minutes on both instruments there. Here's where you can really see some saturation happening. And it is true, like the timsTOF Ultra 2 is tuned for single cells. And so to throw 10 nanograms at it, and especially where we are working at these very low flow rates, so it's even brighter ion source. So the 10 nanograms probably looks more like 50 nanograms. It just starts to saturate here. So, and I think this is a really good story for the proteomics world as well, is when you compare intensities that you observe on the Astral to intensities that you observe on the Ultra, they look pretty good. 0.83 at 200 picograms, 0.86 at 500 picograms, and then you start to see a little bit of saturation effects that drop it down. But isn't it nice to know as a field that these are both good instruments, and the intensities that you measure on one agree with the intensities that you measure on the other. And there's always going to be some spread, right? Because 1% of these are not even correctly identified if you set your FDR to that. Okay, so this is just kind of cool. Okay, so Chow comes to us from the Dalian Institute of Chemical Physics in China. And he's like, well, why aren't we just making our own particles, our own chromatography media? And I'm like, well, because I have no idea how to do that. But he's like, oh, yeah, it's easy. This has been around for 100 years. So here, and not only does he make these particles, but they are beautiful, right? So these are non-porous particles, so you have to worry about the sample loading capacity of these, but not if you're doing single cell proteomics or low nanogram proteomics. You still have plenty of sample loading capacity, but man, I could just pin that up on my wall. Look how pretty those are. And so with this, he has been going crazy fast with the LC. This is not like multiplexed, so it's not like we have so 2,880 samples per day, per se. But what this does show is if we had a system capable of 2.5 minute separations or more interface, you can actually, so this is 200 picograms, 1 minute, 2,800 proteins, 5 minutes or 10 minutes, and then with 2 nanograms, So this is just amazing to me that with label-free, we can start to think about high throughput proteomics without multiplexing. Because like I said, we're bad at it, right? But we can maybe do some fast chromatography to mediate that. Now the CVs are not great at 0.5 or 1 minute here, but they get pretty reasonable at 2 minutes and 5 minutes and 10 minutes. And we think we have some things we can do to maintain the proteome coverage while improving the reproducibility there. And what we found in a hurry is that if we do really fast analyses without updating how we're doing the data processing and analysis, we've just moved the bottleneck one step down without really solving the overall throughput issue. So Xiaofeng, the same guy that did that first 2-column system and the same guy who's basically running Micromics, he was like, what you guys are doing with your data here is just disgusting to me, like moving things on hard drives. So he came up with this system where we have an automatic file uploader that goes to this server, backs things up, can distribute different jobs to different computers, and can provide kind of a simple readout of things. And this is very extensible, right? So right now the functionality is pretty limited, but I just feel like we have these awesome tools out here that we still have to, like, our data are being analyzed by computers, but you still often have to sit there telling the computer what to do. And this is what we're trying to avoid. We want a robot to tell the computer what to do, or a computer to tell the computer what to do. All right. And so this also helps us because it's analyzing the data as fast as it's collected, so we can see if something's going wrong there, and trying to make this as easy as we can. You can see, this is from our we had about 6,000 runs just straight, about 250 plus samples per day. This is back in October. So, and then the MS Connect can just kind of give us a quick readout, seeing how things are going. So hopefully we've shown some advances across the workflow for label-free single-cell proteomics. I think it's becoming easier all the time. Still not as easy as I would like it to be, but we're making progress. We have a throughput right now of about 288 cells per day, about 3,000 or so proteins per cell. And then we have this MS Connect to automate data management, analysis, and visualization. And with that, I'd just like to thank our sponsors. I hope these funding agencies are still there in a year. And thank my wonderful research team. And thank you for your attention.
Great. Well, I'll handle the questions, and I'm going to abuse my power as the holder of the microphone to ask
Yeah.
one myself. So I think the sample preparation that you described is really exciting. We get rid of those steps. We hopefully minimize some sample losses, and so the plots you presented showed that we don't see differences in the number of proteins we see. What I wonder is if we see differences in, say, the sequence composition or the subcellular localization of the proteins by those two different prep methods.
Yeah, absolutely. So we're probably missing, we're not doing anything to decross-link disulfide bonds, right? And so we're probably losing the specific peptides that are involved there. But that's, unless you really care about that, and there are great reasons to care about that. But for global proteome profiling is probably not one of those. So that's okay. In terms of we were concerned about like hydrophobicities and things like that. But looking at the gravy score of each prep method, it doesn't show any bias that way. So
Thanks.
yeah, thank you.
So a very interesting talk. So when comparing the different gradient lengths, you were mainly looking at the total protein identifications, and also I saw coefficient of variation. Did you also look into the accuracy? I saw that you mentioned
Like
you
with
had the mixed species.
Yeah, the mixed
samples
species
there.
proteome.
Did
Yeah,
you-
these are pretty early, so we haven't. Even if we collected that data, I don't know that we've analyzed that data. So it's stuff to look at for sure. Yeah, and we love that human yeast E. coli mixture for that. I even saw it, and you can buy it. You don't have to prep it yourself because there's somebody in Europe that's selling it now. So yeah. Yeah, hi, Yuju.
Hi, very exciting development. I have a question about the peak width. When you convert the retention time in such a short gradient, how will that impact the peak width and the peak shape? And is it okay also for quantitation?
Yeah, so the peak widths, as you go to shorter gradients, the peak widths kind of naturally get narrower, right? Because they just have less time to disperse on the column. And so I'm not sure I followed the rest of the question. Yeah, so that's why we're looking at the CVs, because at some point you're going to be undersampling those peaks, right? And so that's a concern for sure. There are things you can do to ameliorate that. For example, match between runs is one way that you can do that if you get enough MS1 points across the peak. There's also we've also looked at the peptide CVs, and they're not very bad either, which is kind of surprising. But you're rolling up a bunch of peptide measurements into these protein measurements, which I think can effectively give you more points across the peak than you have at the peptide level. But it may also be, because certainly this is going to be a concern, right? If you go to sub-second peak widths, you're going to have to change your acquisition method. But experimentally, we're not seeing, if you have a 15% median CV, that's not that much worse than a typical 30-minute analysis that you would see. So there are other sources of variation, and peak sampling, satisfying the Nyquist theorem and getting enough points per peak is one of those. So I think there are other things in there, but it's definitely something we are certainly keeping an eye on and looking for alternative strategies to ameliorate that as we get faster and faster, right? So yeah, does that sort of answer the question? Okay, thank you.
I think we had an online question as well.
Yeah.
Okay, so enjoy the talk. Why have any particles use ploy columns, no back pressure, ASMS presentation?
Okay, so plot columns probably. porous layer, open tubular columns. Yeah, so we have for a plot column to work well, or for a fully open tubular column to work well, this is where you completely get rid of particles and you just rely on sorbent coating the walls. I think that's probably what they're talking about. So we did one kind of proof of concept paper led by Ying Zhu about 5 years ago. But to get effective mass transfer, you need that capillary to be about 2 microns or less. And so it becomes very difficult to do sample loading there, and your flow rate is about 1 nanoliter per minute there. So all the challenges that we have in going to lower and lower flow rate get really severe with that. So it's been shown that the separation themselves can be absolutely beautiful when working with a plot column. It's the sample loading and the electrospraying that haven't totally been solved yet for that.
And then our second online question says, do you believe in 1% FDR for 2-minute gradients?
Do I believe in 1% FDR?
Do you believe in 1% FDR?
We are relying on tools that other wonderful developers have created. So a lot of false discovery rate algorithms don't rely on retention time at all, right? And so the retention time is essentially irrelevant for a lot of these things. So yeah, it's things where none of these stories are completely set, but there are certainly ways you can experimentally test for FDR to complement the readout of your software, right? With multiple species, decoy databases and things like that. So
right, yeah.
Absolutely.
Okay, and then we'll do one more online question. Where do you foresee single-cell proteomics going in 5 years? Will we be able to do all human proteins, and maybe some challenges that you see in those goals?
Yeah, so I think it's already amazing that there are workflows. The Nikolai's group has shown these throughput of 1,000 proteins or cells per day, and I don't think he would say we're at our limits yet at all, right? And then going a little bit slower with label-free single-cell proteomics, we're seeing now reports of 5,000, 6,000. So about half the proteome is now being profiled in single cells, which is just super surprising. Like that first slide I showed of the 125 cells, that was probably like 300 proteins identified from that 125 cells. So it's really remarkable what the mass spec vendor, developers have provided, along with the, pushing the sample prep and the separations and the data analysis. It's like taking all of that to get to where we are. I don't know, sure, maybe there's a way to get to 10,000 proteins per cell in a few years. I think most of us are more concerned about getting to more cells per study, right? So there's a lot of emphasis on throughput, but you want to have your cake and eat it too. And they're both super important. You can't study a protein that is among those that you didn't see, right? So you just can't, you're just blind to those. So if you happen to be interested in protein X, you better have sufficient coverage that gets that one. So yeah, but I don't know. Yeah, we're excited. We haven't seen anything plateau yet, right?
No.
We are here. This is the 8th of these meetings. We have not seen anything plateau yet, I don't believe. So thank you.
Thank you. Yeah, lots of exciting things to look forward to. Let's give a hand to Ryan.
Thank you.